Today you are going to take Cornell Notes on DNA. It's chapter 12.1 and 12.2 in your textbook. Now remember, for Cornell Notes, the first thing you need to do is you need to draw a line that's about an inch over from your margin. And this should be a straight line. Use a ruler or use the side of another notebook or a binder to draw that line. The title of your notes should go up above, so DNA 12.1 and 12.2. The page number needs to be in the upper outer corner of your page, and then at the bottom left, don't forget to sign and date your pages. And all notes should be in pen. Now, when you start taking notes from the video, from the bullet points that you see, and maybe any additional things that you want to add, you are going to take notes on the right-hand side of the line. Then what you're going to do when you're all done with the notes, whether they're 1 page or 2 pages or 3 pages, you are going to go on to the left-hand side over here, and you are going to write in questions. These are questions that could be answered by what's in your notes, and try and write some higher level questions, questions that compare or contrast, questions that relate to things that you've learned previously in the semester or last semester. And you should have at least 5 questions, if not more, in the left-hand columns. Then on the very last page of your Cornell Notes, not every page of the Cornell Notes, just the last page, you're going to draw a line near the bottom, and you are going to write a summary. And the summary should be at least 5 sentences, and it should summarize in your own words the main important parts about the notes. So what is the genetic material? The structure of DNA was discovered by Watson and Crick in 1953. It was a molecule that scientists knew existed, but they didn't know what it looked like. They knew it passed on information from parent to child, but they had no clue what the structure was. So Watson and Crick were working on this, and they were able to determine the 4 different bases, adenine, guanine, thymine, and cytosine. They knew they were involved, but they didn't know how they were put together. And so they ended up using the work of Rosalind Franklin to help them make their discovery. And this was something that was kind of controversial, at least after the fact.
When there are scientific discoveries, everybody wins, but also sometimes there are a few losers. But few scientists have lost out more famously than the woman who helped discover the structure of DNA, Rosalind Franklin. For that discovery, almost everybody knew the names of the men who got most of the credit, James Watson and Francis Crick, and what people did know about Franklin's contributions, they knew mostly from Watson's 1968 book, The Double Helix. In it, Watson describes Franklin as belligerent, emotional, and unable to interpret her own data. Forget for a moment the unpleasantness of insulting a woman who had been dead for 10 years at the time of the publication of Watson's book, or that he repeatedly refers to her as Rosie, a name she never used. The fact is, had she been alive in 1962 when Watson, Crick, and Maurice Wilkins were awarded the Nobel Prize for their discovery, many believe that Franklin would have, or at least should have, shared the stage with them. Born in 1920 in London to wealthy parents who stressed the value of education, Franklin studied physics and chemistry at Cambridge University. She earned her PhD with a thesis on the porosity of coal before moving to France in 1946. There, she became an expert in X-ray crystallography, a skill that would prove invaluable when she returned to England in 1951 for a job at King's College. Her arrival there coincided with a race among scientists at labs on 2 continents to be the first to deduce the structure of DNA. Franklin and Wilkins worked at the same lab, leading separate research groups, but their work inevitably overlapped as they worked the DNA puzzle. Many scientists then believed DNA had a helical structure like a corkscrew, but it hadn't been confirmed, and there was disagreement over whether it was a single or double or triple helix. Using X-ray diffraction techniques on crystallized fibers of DNA that involved exposures lasting hundreds of hours, Franklin was able to separate patterns that had baffled other researchers. In early 1952, one particular pattern that she would label as photograph 51 clearly showed 2 black stripes, the first real evidence of a helix with multiple chains. The now famous X-ray portrait not only confirmed the double helical shape, but also hinted at its manner of replication. Franklin continued her analysis, unaware that at nearby Cavendish Laboratory in Cambridge, Watson and Crick were working on their own models, but still unable to confirm the helical structure. Though Franklin had yet to publish her images, Watson got a peek thanks to Wilkins, who shared the photograph with his rival in early 1953 without the knowledge or permission of Franklin. Watson wrote of the photo, "The instant that I saw the picture, my mouth fell open and my pulse began to race. Less than 2 months later, using their own data, Watson and Crick announced to the world that they had discovered the structure of the double helix." Franklin's analysis and images would be published in the same 1953 issue of Nature, in which Watson and Crick announced their findings, but by that point, it was a postscript. Franklin left King's College in 1953 to continue her work at Birkbeck College in London. While traveling in the US on business in 1956, she discovered a lump on her abdomen that turned out to be ovarian cancer. She died less than 2 years later at the age of 37. Tragically, her pioneering work with X-rays may have led to her early death. Like many scientists of her time, she rarely took precautions to protect herself from radiation. Over hundreds of hours spent taking images. No matter what anyone said or wrote about her, the world deserved more than 37 years of Rosalind Franklin.
Alright, so the structure of DNA should be familiar to you. We built a model last week and you read about it. So DNA is composed of subunits called nucleotides. And the nucleotides are made up of 3 parts. There's a deoxyribose, which is a sugar, which is the gray part in this picture. There's the phosphate group, which are the blue circles here. And then we have bases. And the bases are made up of nitrogen, so they're called nitrogenous bases. And we have 4 different kinds: adenine, thymine, cytosine, and guanine, A, T, C, and G. Now there are 2 different types of bases, or 2 different types of nucleotides: the purines and the pyrimidines. The purines are the bigger ones, so here it's the green one and the red one. The pyrimidines are the smaller ones, purple and yellow. So adenine and guanine are purines, and thymine and cytosine are pyrimidines. Now, what I would like for you to do is think of a trick that will help you remember the difference between purines and pyrimidines. I'm going to tell you my way that I remember which one's which. I went to school at UC Davis, and our nickname was the Aggies, or Ags. And we started out as an agricultural school. So the way I remember this is I say Ags are pure. So A and G, adenine and guanine, are pure, purines. And so I'm thinking about like, yay, we're like solid, pure sports fans and represent all of our teams. So Ags are pure is my way to remember that A and G are purines, so then that must mean C and T are pyrimidines. I want you to think of your own way to remember the difference between purines and pyrimidines. Now, when Watson and Crick were figuring out the structure of DNA, they used some information that Chargaff had published. Chargaff was a scientist, and he did some work and realized that the amount of adenine always equaled the amount of thymine, and the amount of guanine always equaled the amount of cytosine. And this kind of makes sense, because if A pairs with T, there should be the same amount of molecules when they are looking at a strand of DNA. Same with guanine and cytosine. So I want you to look at your drawing of DNA that you have in your notebook. Using the words purines and pyrimidines, I want you to explain how the bases pair with each other. So not just that A pairs with T and C pairs with G, but I want you to look at it looking at purines and pyrimidines. What is the relationship between the two in the structure of DNA? Next, what is one trick to help you remember that A and T pair up and C and G pair up? All right, so Watson and Crick came up with the model of how DNA is structured. It's a double helix. And A pairs with T with a double bond, and G pairs with C with a triple bond. Now, one strand of DNA complements the other. So the sequence of bases on one side, on one strand of DNA, determine the sequence on the opposite side. It's just like peanut butter goes with jelly, they complement each other, so it does one strand of DNA and the other. And they're held together by hydrogen bonds. So if you look below, we have strand 1, ATC, GAT, TA. What is its complement? What does A pair with? What does T pair with, etc.? What I'd like for you to do is I'd like for you to write out the second strand of this DNA. And you should have wrote that A pairs with T, T goes with A, then we had G, C, T, A, T. And don't forget to draw in the bonds, whether they're double or triple bonds. Now, DNA is found in the nucleus of a cell. DNA cannot leave the nucleus. It won't fit through the nuclear membrane. Now, just a side note, you will find some DNA in the mitochondria of cells. It's called mitochondrial DNA. This is passed on only from mother to child. We're not talking about that DNA. We're just thinking about the DNA, our main DNA, our genetic material found in the nucleus of a cell. DNA contains the instructions for the cell. It tells the cell what to build, what proteins to make. And depending on the type of cell, only certain genes can be expressed. Another name that you might see later is DNA is also called chromatin. Now, DNA needs to replicate, it needs to make copies of each other. So when cells are dividing, when we're making copies of cells, we need to copy that DNA. So before cells can divide, that DNA obviously needs to be copied. DNA replication is semi-conservative. What that means is we have our double helix strand of DNA, and one strand of it goes on to the new cell, and it builds in the new cell a new strand of DNA. And certain enzymes help with this copying. There's helicase, which is an enzyme that unwinds the double helix. And then DNA polymerase brings in new nucleotides.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
Here is a diagram of DNA replication taking place. The red strand of DNA is the original strand of DNA. And you can see that helicase is an enzyme that's moving its way upwards, and it's breaking apart the bonds between the bases, and it's basically separating the strands. It's making them into more of a ladder shape. Now we have the DNA polymerase, which is building the DNA. And you can see here it's building new DNA. But notice it's going in opposite directions. The reason it goes in opposite directions is that DNA always builds from a 5' end to a 3' end. Now, remember we talked about when we made the model that the two sides of DNA are in opposite directions. They're anti-parallel. Well, it has to do with the carbons, the 3 and the 5. So they number them based on which carbon molecule is facing upwards in the sugars. And so here we have this blue DNA. This would be the 5' end, and it's building it from 5 to the 3' end of the blue DNA strand. Whereas this red DNA, its 5' end is on this side, its 3' end is up here. So DNA always builds in the direction of 5' to 3'. And so DNA is being built in opposite directions. Now, if the helicase is unzipping in an upwards direction, this DNA is going in that same direction, and it's going to be a continuous new strand of DNA. So it's called the leading strand. The opposite side, though, because it needs to build from top to bottom, what's going to happen is it's going to be built in small little pieces. So we'll have this blue piece here, then as the helicase continues moving up, it's going to have another little chunk here, then it's going to go up and have another little chunk. So that's called the lagging strand. So how does DNA make a chromosome? DNA coils around a protein called a histone to make what's called a nucleosome. So in this picture here, you can see a nucleosome where we have DNA folding or coiling around histones, and it makes this nucleosome. Then it just continues to build into this shape here that we see, which is kind of the traditional shape of a chromosome that you're used to seeing. Humans have a total of 46 chromosomes, 23 pairs. So you have 2 of each chromosome, which you can see in this picture. You have 2 chromosome 1s, 2 number 2s, 2 number 3s, etc. Then down here, your 23rd pair, those are your sex chromosomes. You either have XX if you're female or XY if you are male. Each cell in your body has every single one of your chromosomes. So what's a gene? Each chromosome has multiple sections that code for different proteins. Each colored section here, it's kind of gray, but each colored section of a chromosome, a band, is a gene. And another name for a gene is an exon. The sections in between the genes, the white part here, those are introns. And originally scientists used to call them junk DNA because they didn't really do anything, but now scientists have determined that there is an actual purpose for the introns.